Okay, let's get started. So, hi everyone. My name is Derek. I'm a business development manager here with Texas. Thank you for everyone for coming today. We're excited to have you here, as well as we'll make sure to post this on YouTube as well. So feel free to rewatch it there as well. The title of today's talk is P53 Mutants, DNA Medicine, and Mobile Genetic Elements. Before we get started, just want to quickly introduce Texas for those who might not be super familiar. So we're committed to advancing biomedical research by providing innovative and high quality animal models that help accelerate the development of next generation therapeutics. Our goal of our company is to help bridge the gap between basic research and clinical application. And we're excited to share today's insights that can help you achieve that. So before we get started, I want to make sure if anyone has any questions during the talk, we have a Q&A chat box. Make sure to put them in there and we'll get to them at the end of the talk. So I'm thrilled to introduce today Dr. Yang Li. He earned his PhD in molecular biology in 1999 from the Shanghai Institute of Biochemistry from the Chinese Academy of Sciences. He followed up his PhD with his postdoctoral training at Yale University here in the US. He has over 20 years of experience in cancer biology. He's had faculty positions at numerous institutions, including the University of Louisville, the Cleveland Clinic, and he's currently at the Baylor College of Medicine in Houston, Texas, where he also is a CPRIT scholar, and he's a member of the Dan L. Duncan Comprehensive Cancer Center. As a fellow of the American Association of the Advancement of Science, Dr. Li has authored over 210 peer-reviewed publications, and he's received numerous honors, including the Achievement Award from the Society of American Asian Scientists in Cancer Research. His research focuses on the tumor suppressor P53, mutant-specific cancer immunotherapies, DNA-encoded antibodies and vaccines, as well as environmental carcinogenesis. Dr. Li leads national and international collaborations aimed at developing novel strategies for cancer prevention and treatment. So without further ado, let's get started. Dr. Li, the floor is yours.
Thank you, Derek, for the nice introduction. Today, I'm going to talk about three topics. The first one is, let me see why it's not moving. Okay, the first one is how to use antibodies targeting P53 mutant. The second is the biological drugs, including the DNA plasmid. The third one is the mobile genetic element in cancer and aging. For the first one, I would like to tell the tale of two tumor antigens. You're probably difficult to recognize the name. On the right is official gene name called MS4A1. I thought there are many people can tell what this gene is, but I tell you the nickname. It's CD20. It's a B-cell specific antigen, and it's discovered as a B-cell antigen using a monoclonal antibody in the 1980s, and it was studied as a B-cell antigen. And so on the right, actually, it's called P53. At one point, they are called a tumor suppressor, tumor antigen P53, all called NYCU13. Basically, it's from Dr. Old, in New York, they found a lot of antigens. The NY is the New York Institute, then the CO is the colon. The 13 is the number 13 antigen in colon cancer. So originally P53 is discovered as an antigen. It's combined to SV40 large T antigen, as well as, in addition, it can elicit the antibody response in mice and rabbits very strongly. And that's before 1980. It was studied as a tumor suppressor, oncology, and a transcription factor. So what's the difference is on the right, this CD20, or you can call the official gene name MS4A1, it was studied as an antigen and targeted as an antigen. There are now 26 antibodies approved by the FDA to target CD20. For the one P53, it was studied as tumor suppressor oncogene, or mutant form is oncogene, then a transcription factor. it was targeted as the oncoprotein P53 mutant. However, there's no drug approved by the FDA. Why there's no drug target the number one mutated gene in cancer? There are many reasons, depending on how you view it. You know, tumor suppressor overall is difficult to target. There's no drug target tumor suppressor. And oncology, regardless of KRAS, EGFR, there's lots of drugs. So the difference between P53 mutation and KRAS mutation is KRAS, when it is mutated, you still have the KRAS protein. It's just a supercharged KRAS protein, ATPase. But P53 mutation, once it's mutated, the conformation totally changes. It becomes a well-folded protein to a denatured protein. So in that sense, you could view the wild-type P53 as an egg. It's a live creature, right? It can get out of the chicken. But the P53 is a fried egg. It is difficult to put the fried egg back into the real egg that can turn into a chicken. And in another sense, you can argue is you try to use small molecule to react the P53. It's like you have a wild-type P53. It's a well, built building, and P53 mutation is a very important element, like a beam in the building. It got something wrong, the building collapse. So you want to use small compound to reactive the P53. It's like you saw a collapsed building. You try to see, I'm going to give one, break or something. I know it's because of the beam problem. But I just want to use a very similar, like a brick or some other suffixed structure, without taking that beam out, defective beam out, and I put this structure and make the building folded to its original shape. That's how hard to target P53 mutant. In another sense, to target P53 mutant is like target beta amyloid lesions in Alzheimer's disease. It's very challenging, but there are, of course, some antibody approved to treat Alzheimer's, but the effectiveness is limited. So we thought about using antibody to target the P53 mutant. Even in the early 90s, people found, the cancer patients, indeed, they secrete antibody in their blood. They have antibody in their blood to target the P53. And on the left, it's a cancer patient. You can detect their serum anti-P53 antibody 600 days before the cancer diagnosis. So even cancer become, overt, cause problem for the patient, the anti-P53 antibody is there. That's serum anti-P53 antibody. Recently, there are other type of antibody. We call it antibody from tumor infiltrating B cell and plasma cell. We call it TUB. Within tumor, there are B cells and plasma cells. They secrete antibody. This antibody can target a variety of antigens, including P53. So how do we take advantage of the natural immunity against the P53 to turn into a drug? So what we know, P53 mutation it's very diverse. There are over 3,000 P53 missense mutations. Primarily focus on a few hotspots like R175, R248, R273. In addition, you have 282, 245, and 249 residues. So for these hotspot mutation, we developed the antibody target 3 of them. We target R175, R282W, and E285K. In the first antibody development, we developed antibody that target this mutated residue. So basically, the epitope is the new epitope, contains the mutated amino acid. So if you have a wild type of amino acid in the peptide, the antibody cannot target. But of course, you have to develop one antibody for each of the missense mutation. And we developed against the three mutant residues, and one of them is the E285K as shown here. After the E mutated to K, we have antibody just targeted that mutated amino acid. And we have this antibody with a pretty decent KD value, less than 1 nanomole. And we purify the antibody, the binding to the antibody is shown here. It's effective to target the E285K, but not other antigen. In addition, you can see the specificity. If you express wild-type P53 and the R282W, the antibody do not have the signal. But if you express the E285K P53 mutant, you can detect the signal very strongly. So next, we use a cell line that it's a mouse syngeneic cell line, MC38. It contains endogenous mouse P53. We knock out the mouse P53 allele and overexpress the human P53 E285K. This cell line, we then use this antibody to treat. So the first type of treatment, we do not use the purified antibody. We use lipid nanoparticle to packaging a DNA plasmid, actually 2 DNA plasmids. One expresses the heavy chain of the antibody, the other one expresses the light chain of the antibody. We can do intratumor injection of the lipid nanoparticle into the tumor, and where the plasmid can express the heavy chain and light chain antibody to get this the full-length recombinant antibody expressed in both tumor cells and immune cells. So the lipid nanoparticle in this case are based on the Moderna mRNA vaccine, as we know, against the COVID. So for the first set of experiment, we inject the tumor into the C57 mass first, and after the tumor is established, we treat with the LNP containing the antibody plasmid. And you can detect the antibody in the mouse serum. In addition, you take out the tumor and check the expression of the antibody because the antibody carrying a human FC. You can detect the antibody. It can express both in immune cells, CD45 positive cells, and in the tumor cells, it's the CD45 negative cells. And also, we found that by doing the intratumor injection of the LNP, the tumor growth was significantly inhibited, and the animal survival get improved by the treatment. The second one, we performed the IV injection of the tumor cells into the C57 mice, and then we treated the animal with IV injection of the LNP. As you see here, the metastasis of the MC38 get significantly reduced, and the animal survival improved. In addition, we performed orthotopic injection. Actually, it's not exactly into the colon. We do the IP injection of the tumor cells into the peritoneum of the, including the some of them, including the small intestine and the large intestine. And then we perform IV injection of the LNP. And what you can find is similarly the tumor growth gets significantly inhibited by the LNP treatment, and the animal survival is improved. So in addition, we use a CD34 humanized animal model to test the activity of this antibody against the human cell line endogenously express the P53 E285K antibody. Here we have 2 cell lines express this E285K. One is the BT474 breast cancer cell line. The other one is a multiple myeloma cell line. Both in both cell lines, the antibody with the LNP within the LNP is effective to inhibit tumor growth, but obvious the activity against the BT474 is more significant. Next, we studied the mechanism why the antibody can inhibit the tumor. We we find out through a single cell sequencing and antibody blockade against different immunocells. What we found, there are two potential mechanisms why the antibody is effective even for this P53 mutant an intracellular target. Is number one is it can recruit immunocells like NK cells and macrophages into the tumor, where upon the binding of the antigen and the antibody, the many immunocells express the Fc receptor can binding to the IgG plus the antigen. Upon binding this triple protein complex, the immunocell will be activated. If the immunocell is a dendritic cell, it will endocytose the complex and digest the mutant P53 for cross-presentation. That's the second mechanism. So the antibody can enhance the dendritic cell antigen presentation and further enhance the T cell activity. We also found that during this process, an intracellular IgG receptor called TRIM21 is involved. So if you knock out the TRIM21 in the tumor cells, you basically eliminated the anti-tumor activity of the antibody. So in addition to our work, we use IgG1 antibody format. A group from Duke, they use dimeric IgG1 antibody. What they found is you can use dimeric IgG to target the intracellular mutant target, including KRAS, G12D, and IDH1 R172H. So this process is dependent on the dimeric IgG antibody, dimeric IgG receptor expressed on tumor cells. This receptor is called a polymeric immunoglobulin receptor. We call it a PIGR receptor. So the process is, the tumor cell express receptor upon binding with the dimeric IgG antibody who collab with the antigen. This complex will get inside the tumor cells. It can bind to the mutant protein like KRAS G12D. There is a transcytosis allow the target mutant protein get outside and to enhance anti-tumor activity and antigen presentation. So we tested if our antibody, if we make it to the IgG format, can it help to inhibit tumor? That's what we did with IgG format. We do the similar treatment with similar, the subcutaneous tumor. With the intratumor injection of the lipid nanoparticle contains the plasmid to express the mouse dimeric IgG. What we found that the treatment is effective to inhibit the tumor, and also this process is dependent on the PIGR receptor. If we knock down the PIGR receptor, you can see this anti-tumor activity get reversed. Of course, if you knock down the PIGR receptor, the animal survival will go back to the untreated or control treated. So going to the next stage, we want to develop antibody target multiple P53 mutant. So the idea would be, go back to the basic about the KRAS mutation and the P53 mutation. KRAS mutation, if you develop a small compound, you have also to develop one small compound for each of the mutant. Of course, someone is claim they have this so-called pan inhibitor, but in real life, you have to develop one small compound inhibitor against the G12C. It's approved by FDA. Or the G12D is ongoing research and clinical trial. It's not approved yet. So for the P53 work, there are many groups develop individual small compound targeting Y220C, or we develop small compound, we develop a monoclonal antibody targeting E285K. So we think about, because the mutant P53 have a common feature is after mutation, lots of P53 mutant lose their native conformation, become unfolded. So can we, check if there are cryptic epitope can be targeted by an antibody? So basically, you have lots of different mutations. Many of them we call protein, P53 protein unfolding. The unfolded peptide can be cryptic epitope for antibody binding. So in the native form, this antibody cannot bind to the P53, but upon P53 mutation, many protein, P53 protein will be denatured. Some of the denatured peptide could serve as the antigen for P53 antibody development that allow, target multiple P53 mutation. So that's what we found in the P53. There is a peptide involved into the DNA binding to the P53 in the minor groove of the DNA double strand, right? So this peptide is well folded in native P53, but once the P53 mutated, this peptide is denatured. We develop an antibody that targets the denatured peptide. We call it a cryptic peptide within the P53 loop 2. And we have the mouse antibody. We further humanize this mouse antibody into a humanized antibody we call the P20. And so this is the data we have for this human antibody. On the left is the imaging. We label this antibody with Psi5.5. And then we inject different tumors into the immunodeficient mice. Once the tumor formed, we inject the antibody with red fluorescent, as you see here. The P20 antibody can target two P53 mutant, R175H and R273H. We have R175H monoclonal antibody can only target the R175 mutant cell line. And on the right, we perform subcutaneous tumorigenesis, and then we treat these tumors with the two type of antibody, as you see here. So in the P53 knockout cell line, without the P53 gene, the antibody is not effective to inhibit the tumor. For P53 wild type cell line, the antibody are not effective. For the P53 R175 mutant cell line, both antibodies are effective, even though our P20 is more effective than the R175 specific antibody. However, for the R273H mutant, only our P20 antibody is effective to inhibit the tumor. This data suggests that the P20 antibody can target multiple P53 mutant. So now I would like to talk about the second topic, DNA encoded medicine. As we know, the success of mRNA COVID vaccines is dependent on three elements. One is lipid nanoparticle. The second is within the mRNA, you have a modified nucleoside, allow stable expression as well as reduced innate immunity. The third element is there are two amino acid mutations, two other amino acid mutated to protein mutant to make the spike conformation in the pre-fusion stage. So the antibody you elicit can target the viral infection before the infection rather than post-infection. So in my laboratory for the past 5 years, we want to develop the nucleic acid medicine. How do we view the mRNA COVID vaccine? We view the mRNA COVID vaccine as a type of drug, contain the instruction, so genetic codons in either in mRNA format or DNA format, allow the patient themselves to make the active pharmaceutical ingredient themselves. So in this case, you can use the mRNA as the codon or use the DNA as the codon. So this is very important. You think about the drugs, right? The small compound drugs. Of course, patient cannot make the small compound drug. You have one compound that you give it to the patient can do some metabolite can metabolize the compound, make it smaller or add some elements. In some prodrugs, you indeed have the patient to modify the prodrug to make it the active drug. But that's different from the instruction from genetic codon. So today, I would say, if you have a compound, you want to do 100 modification, the price will be significantly higher than you have a peptide. You want to encode the peptide by DNA or mRNA, you make 100 modification on the peptide. So the price of using genetic codon to instruct the protein production is cheaper than the small compound. On the left is a small compound approved in Japan. You need 100 steps of chemical synthesis. But for the protein encoded by DNA, you can see the price now is very cheap. It's less than 10 cents per base pair, and you can prepare DNA plasmid with $20 or $200. $200 you can outsource it. And you do a full-length plasmid sequencing only costs $10. The plasmid DNA or even mRNA can be delivered by electroporation or lipid nanoparticle. The patient make their own drug. Of course, very often these drugs are made with the patient-specific post-translational modification. So this is a new topic, in terms of drug delivery. There's the biosynthesis stage. So what protein can be delivered by the mRNA or DNA? First, you have to think about, for the DNA or mRNA delivered as drugs approved by FDA, what's the dosage, right? What do we know? The lowest dosage you can imagine, there's a third successful COVID vaccine is from Novavax. In their protein-based vaccine, the recombinant spike protein is only 5 micrograms. This is the low-hanging fruit, right? Then going up, you have a peptide hormone approved by FDA. Of course, insulin, as we know, general injection patient with 10 units of insulin, about 0.35 milligram. And you have the GLP agonist like dulaglutide. What we know is FC-based protein. It's 1.5 milligram. You go up to, of course, cancer vaccine not approved by FDA yet, but from Dana-Farber clinical trial, they use synthetic long peptide, 0.3 milligram per peptide, up to 20 of them. So the dosage is about 6 milligram. And you further go up, there are bispecific antibody targeting myeloma. It's talquetamab, 30 milligram, pembro targeting PD-L1, 200 milligram, and atezo targeting PD-L1 is 800 milligram. Of course, the COVID antibody, you need 4.5 gram per injection per delivery. And on the top is the human serum albumin. As we know, you need a lot of them. Very often patients in their terminal stage, they were given the serum albumin at 10 gram, 30 gram, 50 gram, or even. So you can see for the FDA-approved protein as a drug, you have a range from 5 microgram to 50 gram. Of course, we know that the 5 microgram is already successfully sort of countered by the mRNA format, right? The Moderna and BioNTech, Pfizer vaccine, in terms of mRNA level, it's 30 microgram and 100 microgram, but we don't know the exact how many microgram of the spike protein they produce. For the cancer vaccine, in addition to the synthetic peptide, long peptide, Moderna and BioNTech, they have their mRNA version of the new antigen vaccine. The dosage is 25 microgram in the format of mRNA, and 1,000 microgram in the format of mRNA. So at the same time, there's a company using DNA to encode the new antigen. Their clinical trial is equally successful. The DNA dose used is 1,000 microgram, so it's 1 milligram. And of course, we know using DNA, there's a company called Inovio. They tried during the COVID era, they used DNA plasmid, 2 milligram of DNA plasmid as the antigen against the COVID. But this vaccine is not successful. It's not approved by FDA. So what's the limitation of the mRNA DNA? I would say is why we cannot use mRNA or DNA to deliver insulin, because for insulin as a drug, you cannot deliver too much. So basically, you deliver 10 times the normal dose, the patient will pass out. And but from the COVID vaccine, we know the challenge is this drug company did not report the plasma level of the spike protein, the antigen. They did not report the antigen level. But eventually, this antigen stimulated the body to generate a neutralized antibody. This antibody level can vary over 100 times within the, from patient to patient. So there's a big variation, but that does not mean the antigen level vary 100 times. The antigen level could vary maybe just 200%. That's probably enough to deliver lots of other protein drugs. So what our lab for the last few years, we tried to use the existing knowledge of why the mRNA vaccine is so successful. We want to make the DNA medicine successful. We want to have a better lipid nanoparticle. We also want to modify the target to allow better target delivery. For example, if we deliver vaccine, we want them go direct to the dendritic cells. The third one, we want to modify the vector of the DNA plasmid, make a better vector, smaller vector. So the first point is the formulation of lipid nanoparticle. As we know, the first lipid nanoparticle included drug is the siRNA drug, and it used the first generation. All the lipid nanoparticles so far using the 4 lipids. The first generation, the most important lipid is the cationic lipid. In the first generation lipid nanoparticle, they use mcDMA as the cationic lipid. The second generation for the 2 mRNA vaccine, they either use SM102 or the ALC 315 as cationic lipid. So are there better cationic lipid for mRNA delivery? The answer is yes. Actually, one of the most successful lipid nanoparticle company in Canada, they have developed the first generation so-called MC lipid, and they developed the second generation. Now they have the third generation lipid nanoparticle can deliver mRNA about 3 times the efficiency of the second generation. As we know, the second generation is 8 times more efficient than the first generation. So we are interested to obtain this third generation cationic lipid and for DNA delivery. And the second one is you can modify the antigen for better vaccine. For example, this group, they use nanobody targeting the MHC2 as fuse the nanobody against MHC2 to the spike receptor binding domain. What you observe is a significant improved immunogenicity using this fusion protein rather than the standalone ribosome binding domain of the spike protein. This is the second modification we're working on. The third modification we believe is because of the DNA vector. Why the DNA COVID vaccine Inovio INO 4800 failed is that modern plasmid, as we know, come from PPR322. It was found 1977. But this plasmid is for E. coli only. You cannot use it to deliver your target, your payload to human cells. Then around 1996, Thermo Fisher get this plasmid called PVX1 and can do the eukaryotic cell expression. And then a little bit later, there's a DNA vector called pcDNA 3.1. This is by far the most widely used vector for mammalian cell expression. And it got a very decent expression level. Then there's a plasmid called GWAS, what we use in our lab very frequently. And it's an improved version of pcDNA. And of course, there are some more specialty plasmid, one of them called nanoplasmid, very small. So if you put a reporter in this 4 plasmid, what are their expression levels? So what we found is, the vast majority of DNA development, DNA vaccine development, use this PVX1 vector, which is a very lousy vector. So if you put a reporter, you found the expression level is lowest among the 4 plasmid. Nanoplasmid better than GWAS, GWAS better than pcDNA, PVX1 is at the lowest for target expression. And it's worse than, many people use the pcDNA, but the DNA vaccine researchers didn't realize this PVX1 is so much worse than the regular pcDNA. They only use it because the vector size is small, but in terms of expression, there's a lot of element in the vector cause its lower expression. So regarding the different vectors, I would like to emphasize regular plasmid like PVX1 or GWAS or pcDNA, you have at least 3 elements, right? One is the antibiotic resistant gene in E. coli. The other one is E. coli DNA origin, the origin for DNA replication. You want to produce the DNA, amplify it in E. coli. The third one is the real one, the expression cassette for human cell expression. This is what we call the payload. The nanoplasmid is optimized these 2 elements. In PVX1, this antibiotic and the origin count for 1.5 kb. But in nanoplasmid, they use alternative to antibiotic selection. They use RNA-based approach to bypass the antibiotic selection. Then they use a smaller origin of DNA replication, R6K. And this allows a significant reduction of the bacterial backbone. Of course, there's a company called GeneScript. They use even smaller backbone. They just add this R6K and plus the human expression cassette. So what we take this, we compare either GWAS in our lab, compared to the nanoplasmid, the gene circle, we put luciferase there. What we found is quite striking is the nanoplasmid and the gene circle methods allow more than 10-fold improved infection for our reporter gene. In addition, there is a company in England. They just even shorten that R6K, this about 300, 400 BP to about 50 BP. So they have a totally synthetic double-strand DNA, they call it doggy bone. And this is further reduce the non-essential part of the payload. And we are acquiring this technology, and we will compare how this one, the doggy bone DNA compared to nanoplasmid. So go back to the individual target in my lab. We have this on the bottom is the low-hanging fruit like COVID, then peptide hormone, then have the cancer vaccine, then have the antibody. So in our lab, we actually develop a very rudimentary vaccine and using the DNA platform. we found they are effective to generate a neutralized antibody. Of course, we didn't do anything further because this is just a proof of concept. The technology is definitely sufficient to develop vaccines. For the peptide hormone, we are working on to deliver the dulaglutide using DNA. Then for the cancer vaccine, we target a few of them, cancer, it's a tumor associated antigen like GPRC5D with P53, PD-L1, and we are working on PSMA in prostate cancer and other target. And for the antibody, we use the DNA to deliver P53 antibody, including the R175H, R282W, and the E285K. So how hard for patients to make the protein? We know the protein range, the dose is 5 microgram to 50 gram. How hard for patients to make their own drug? What we know is a single human cell contains about 10 million ribosomes. One ribosome can produce 2,000 protein per second. That would translate to about 175 million protein per day. So if you count average protein in human cell is 50 kilodalton to make 1 milligram of this protein, how many human cells are needed to work a full day at the full speed? The answer is 7 cells. Not a lot of cells. As we know, human, we have 37 trillion cells. You just need 7 cells to produce 1 milligram. Of course, you argue it's not a full speed. This 7 cells need to work at full speed, but you can say, oh, maybe 7 cells work 10% efficiency, or like you need 70 cells or 1% of efficiency. You need 700 or even 1,000. 0.1% of efficiency, you need 7,000 cells, not a lot of cells. So the true limitation in our reading and estimation, regardless it's DNA or mRNA, is how to get the nucleic acid to the ribosome. What we know is for mRNA, you deliver 100 mRNA in the cells, only about 1% of the mRNA reach the ribosome. And for DNA, you have 100 DNA into the cytoplasm because you need to go to the nucleus, get transcribed into mRNA, and mRNA get out into the cytosome to meet the ribosome. That percentage is less than 0.1%. So we still have a long way to go to fully realize the potential of codon, genetic codon instructed drug biosynthesis and delivery. So this slide, I want to show the comparison between mRNA and DNA. mRNA is generally delivered by lipid nanoparticle. It allows higher expression, and its transient nature allows better antibody response. The DNA is delivered by lipid nanoparticle or electroporation. It has lower expression than mRNA, and it is more stable. So it's better suited for T-cell response. The third topic I want to talk about the ritual element, the mobile genetic element in human cancer and aging. Recently, there is a significant development in the human longevity research. There's a lot of people, rich people, founding biotech companies. Their major goal is through partial reprogramming to rejuvenate cells and tissues, to reverse disease and treat disease and to delay aging. So the method is based on reprogramming discovered by Yamanaka back about 20 years ago. So you can use 4 transcription factors. You can turn a somatic cell, primarily skin cell, into induced pluripotent stem cells. And about 10 years ago, a group in California, a Belmont group, they partially reprogram the animals. So they just give us very short, brief pause of this full transfer factor into mice. These mice are carrying a premature gene. So doing this partial reprogramming allow the animal survival extend by 30%. In addition, when they take the major tissues like pancreas and muscle, they check the biological clock in these tissues. They found these tissues are getting younger by the treatment, the partial reprogramming. However, as a cancer biologist, we know the cancer is the most significant risk factor for this part of this partial reprogramming. Because what we know is even back in the earlier days of reprogramming research, we know if you delete the P53 gene, you increase mouse partial reprogramming 200-fold, increase the efficiency 200-fold. For human cells, you increase the reprogramming efficiency 50-fold. So this would suggest, if you apply the partial reprogramming into adult patient or even elder patient, you could easily select P53 inactive cells, and you would significantly enhance cancer risk in this patient. That's a big problem. So what I view is, how do we improve human lifespan? What I want to do is, first, I want to review why human lifespan get significantly improved since industrial revolution, right? Since 1800. What we know, there are multiple reasons. You have public health improvement, you have medical advancement, you have improved living standard, and you have reduced infant and child mortality. So overwhelming of these improvement are indeed reduce the infection from bacteria, virus, and yeast and other infectious pathogens. So reducing infection, I would say, contribute the most to increase the human lifespan since 1800. And even in today's world, you check the cause of death in chimpanzees, most close to human in terms of genetics. And you also check the cause of death in human living in caves, so-called hunter-gatherers. Their cause of death, vast majority of them caused by infection. Okay, of course, you can see chimpanzee and the human living in caves also have some kind of violence against each other as the cause of death. So in this regard, it seems reducing infection is from exogenous pathogens is the leading cause for increased human lifespan. My question is, will you inhibit the endogenous pathogens within our genome, increase our lifespan? So these endogenous infection agents are called ritual element. There are many ritual elements in our genome, accounting for like 45% of our genome. Among them, line 1 account for 17% of our genome. Human endogenous retrovirus account for about 8%. So we first focus on line 1. We know line 1 is it's a ritual transposon, and it encoded by one messenger RNA. It can produce two proteins, ORF1P and ORF2P. And in our genome, there are 500,000 copies of the line 1 genes. Most of them are truncated, but there are at least 100, maybe 80 to 100 are full-length copies in everyone's genome. The why line 1 is alternative target for partial reprogramming, because the same group, same California group, they found in the premature mouse aging model, if you deplete line 1 RA, you could expand the lifespan by 30%. So this suggests line 1 is an alternative target for longevity. In addition, line 1 is activated by epigenetic alteration during aging from low-end organisms like worms to Drosophila to zebrafish to mouse and to humans. And the line 1 activation causes genomic instability and chronic inflammation. That's what I consider these are the endogenous infectious pathogen within our genome. So what's interesting is line 1 is also expressed in cancer. About 47% of all cancer tissues is ORF1P positive. In the cancer patient plasma, 5 to 70% of them have this ORF1P. And particularly for ovarian cancer, you can detect about 70% of them. And colon cancer, at least 55% of them. In this case, if you target line 1 for longevity, it will have the advantage, as we know, there are so-called meta hallmarks of aging and cancer. So you take a look at the hallmarks of aging and cancer. There are several elements. It's shared between aging and cancer. The first 3, genomic instability, epigenetic alteration, and chronic inflammation are shared. So, but if you use partial reprogramming, you would have increased risk of cancer because you increase the stem cell stemness, you may enlarge the telomere, you may prevent the cellular senescence. These are all could be the tumor promoting events. So on the other hand, the line 1 inhibition can do against cancer and against aging. So that's what we did. We developed a mouse monoclonal antibody targeting the human ORF1P. Again, here we use MC38 to express the human ORF1P. What we have a couple of antibody showed the tumor inhibition in this MC38 syngeneic model. Next, we humanize this antibody in. So after humanization, what we found, the antibody affinity dropped significantly with a KD of 30 nanomole. We further improved the humanized antibody by affinity maturation. Their KD value get reduced to 2 nanomole. And then we test the antibody activity against tumor cell line carrying the human ORF1P by itself or through the combination with the PD-1 blockade. And what we found, either as a monotherapy, the humanized antibody, here is the 2 humanized antibodies, CR21 and CR13, both can inhibit the tumor, along with their parental antibody, mouse antibody, 4281. And with the PD-1 blockade, the tumor inhibition is stronger. So finally, I would like to pay tribute to the parable of 6 blind man and the elephant. We scientists very often view the biology without the full picture. So, but everything is like you struggle in the dark to pick up one target and for your research. So for the P53 mutant, I view it as a fried egg. So I view a fried egg cannot be easily turned back to a tumor suppressor. So my goal is to use antibody to clear out the mutant P53 in tumor. For the second view, I view the mRNA vaccine as a drug. You just provide the genetic codon as an instruction. The patient can make their own active pharmaceutical ingredients. Regardless of the vaccine, hormone, peptide, all the antibodies, you can always use the instruction allow the patient to produce the drug themselves. The third one, I view the human longevity. As far as we know, our improved lifespan is because you reduce the infection from exogenous pathogens. To moving forward to further improve the lifespan, you may target the endogenous retro genetic element in our own genome. That's why we develop antibody to target the OFMP, and we believe it may have some anti-tumor activity as well as the promote the longevity. So finally, I would like to thank the funding agency and the people in my lab who are hardworking and dedicated to cancer biology and therapeutic development. Thank you all for your attention, and now I welcome any questions.
Thank you for the great talk, Dr. Lee.